Hi guys, today we're going to talk a little bit about prokaryotes. So this is chapter 19, and I know you've already done some work on viruses, so here we go. Let's jump right into prokaryotes. So one of my favorite questions that you guys already know about is compare and contrast. So this is the beginning of chapter 19 talks a little bit about the differences between archaea and bacteria. So what I've done here is really put down some basic ideas about some differences and similarities between the two. But I'd really like to have, when you read the section, to add to this. Clearly we know that they're both prokaryotes. Let's just get this off of here. So they're both single-celled, and because of their prokaryotes, they lack membrane-bound organelles. They're both small, but you're going to find that the sizes are actually different between the two, so that I would encourage you to get some of that information. They were both reproduced by asexual reproduction, which is called binary fission, and here are some differences between those two different groups. So as you read the text, it's really important to kind of add to this Venn diagram. So in your notes, I would recommend creating this Venn diagram and adding to it when you do your notes, so that you have a good study guide to talk about the differences and similarities between that if you were to get an essay question on that. The next thing is their structure. And although there's not a classic picture in our textbook in chapter 19, there is on page 73 to 74. So I would recommend coming back to that, because that's when we really started to talk about cells. And on page 73 there's a really nice figure, figure 419, that actually shows you something very similar to this. The only thing that this particular picture doesn't have that the page 73 actually does, and let me try to actually draw that in here, is a plasmid. Oops, is it going to let me do that? I guess not. But anyway, a plasmid is a circular extra piece of DNA that some bacteria have. So it's not part of their nucleoid region, but an extra little piece of genetic information. So when you go back to page 73 and 74, you're going to look at this figure, which I would recommend. Make sure that you're comfortable with all of the parts of the bacterial cell, and not only are comfortable with identifying each part, but you can guess about what I'm going to say next, and that's actually the function. So what is the function of the nucleoid? What is the function of the capsule? What's the function of the cell wall? What's the function of the cytoplasmic membrane, which is really just another name for the plasma membrane? A lot of these you're already familiar with. The ribosomes, we know that the ribosomes are the site of protein synthesis, and the flagella is used for locomotion. But some of these other terms you might not be familiar with, and I would again go back to page 73 and 74 and make sure you know the function of all of these particular items. So that's just a little refresher on the prokaryotic structure. Now prokaryotes, we just finished the section on classification. So when we talk about the King Philip came over for good spaghetti, that was the classification and kind of structure, the hierarchy about how we classify organisms. And bacteria, whether they be archaea or eubacteria, are both organisms that we would be interested in classifying. And we not only do that with their genetic material and some other properties, but we do that in other ways. And so these are the classic ways. And so you could make a mnemonic device with this, and you might want to just pause the program to come up with a mnemonic device. Unfortunately, this doesn't spell anything, and it doesn't have to be in this order, but this is how I'm going to go through the presentation today. So if you know that they're classified by their shape, their locomotion, staining, nutrient requirements, and pigments, that's an important piece to this topic on prokaryotes. So let's actually get into the first one, their shape. So bacteria are classified by their shape, and we've already done these prefixes, and the reason why I did them before is so that when we actually got to this chapter, this particular word would sound familiar to you. So there's three types of shapes. There's round bacteria, there's rod or a little stick bacteria, spirillum, which is spiral. So those are the three shapes that we have. So here again, the coccyx, which is round, bacillus, which is a little stick, and spirillum, which is spiral. So that's the second term in the word of how we would classify the bacteria. The first is how they are actually clustered, or how they actually organize with one another. So if I said Diplococcus, that would mean that I have two spheres, two round shaped bacteria. So I would say Diplococcus. I could say Staphylococcus, or I could say Streptococcus. So Staphylo is like a cluster, Strepto is in a chain. So the other things that we could have a Diplobacillus, Staphylobacillus, Streptobacillus, or I could have a Diplospirillum, Staphylospirillum, or Streptospirillum. So the first word is how they're arranged. The second word is how their shape is. Diplo is two, Staphylo is in a cluster, Strepto is in a chain, and then these circles, rod shapes, or little stick, and then spiral. So let's actually see if we can name these. So as you look at these, try to figure out what you would call them. Are they a Diplobacillus? Are they Streptospirillum? So why don't you pause the program and see if you can identify these particular slides, these microbiology slides, and then come back and we'll see how you did. So how'd you do? Well, clearly these are circles, right? So these would be a coccyx. And how are they arranged? Are they in a line? Are they in a cluster? And they actually are in a cluster, so this would be a Staphylococcus. And if we look at these really are in a chain. They're not really in a cluster per se. So this would be Streptobacillus. So this is Streptobacillus, and this is Staphylococcus. So hopefully I'll be making microbiologists out of all of you guys. The next way is locomotion. So prokaryotes actually move by different methods, and so these are just different pictures of all the different combinations that prokaryotes can have. They can have one flagella, they can have two, they can have many, they can have like crazy hair days, and their movement depends on the particular type of bacteria we have. They can squirm, glide, propel themselves. The flagella that bacteria have are made out of this particular protein. If it ends in IN, it's flagellin. IN means it's made out of a protein. And bacteria flagellum does a rotating movement versus archaea, which moves a little bit differently. So here I've got a little note to help remind you that there may be some differences between archaea and bacteria, and you should feel comfortable with those differences. And if you really were curious, on page 357 there's another figure, and you know how much I love figures and tables, that highlights those differences. So if you want to take a moment right now to pause the program and go to page 357, I would encourage it. The next way the bacteria are classified are by their staining. So gram-positive bacteria have a thicker peptidoglycan cell wall layer. These are pictures of the cell wall of a gram-positive bacteria, and these are the cell walls or cell membrane and cell wall of a prokaryote that stains gram-negative. So gram-positive bacteria have this cell membrane, this plasma membrane here, but then their cell wall has a lot of peptidoglycan. So when we stain this particular bacteria will stain purple. And so if it's purple, it's a gram-positive bacteria, and again it has a thick peptidoglycan layer. On the other hand, on a gram-negative bacteria, if we stain this, it's only got a very, very thin layer of peptidoglycan. Still has the cell membrane layer, much like a gram-positive does. The peptidoglycan layer is very, very much smaller, I should say. And then it has this extra layer on the outside. This extra layer tends to be made out of more lipids, and you can see here it's this lipoprotein layer, and this layer actually is a protective layer for the bacteria, and it's very, very difficult to treat gram-negative bacteria if we were to be infected by them. So people tend to get more sick with gram-negative bugs, and they're also require different antibiotics in order to treat them, and so the antibiotics tend to be pretty strong as well. These, if we were to stain, come out pink. So if you were to go to the doctors and they were to culture, say, your throat and put it on a petri dish and grow it and then look at it under a microscope, you could say, was it a gram-positive or gram-negative bug, and that tells the doctor about what these cell walls are actually made out of. Do they have this extra layer which is difficult to treat, or is it a gram-positive bug that has only 2 layers, a peptidoglycan layer and the plasma membrane layer? So if we go back to our pictures, with these pictures, would they be gram-positive or gram-negative? So go back to the other slide if you want, just pause it for a second and then come back and we'll see how you did. And if you said they're both gram-positive, you would be correct. So we not only can say that this is Staphylococcus, but it's a Staphylococcus gram-positive bacteria. This would be a Streptobacillus gram-positive bacteria. Again, the more descriptors you have, the better communication that you could have with other people about that particular prokaryote. One of the last ways that bacteria are classified is by their nutrient requirements, or the nutrients that they require in order to grow and live. So there's 4 different types, nutritional types, and here they are listed here. And we already know that photo means light, auto is self, and troph is feed. So this is a self feeder that uses light as their energy source. So anytime you see a photo, you know you have your energy source as light. If it's an autotroph, it will always use a carbon source of CO2. So here we see an autotroph here, that uses a carbon source of CO2. So again, anything with photo, energy source is light. Anything that says autotroph, the carbon source is CO2. So those are some easy ones to remember. Now chemoheterotrophs are just like us. We use organic compounds for our energy source, and that's our carbon source as well. So this is kind of where we fall into the food chain. On page 359 in your textbook, it talks a little bit about nutrient requirements as well. So prokaryotes, actually, we said this at the beginning, reproduce asexually, and the process that they go by is binary fission. And this actually can go so quickly if conditions are good, and that's why you can get really sick from a bacterial infection pretty quickly, because the bacteria, if the conditions are good, will just go like wildfire and reproduce really quickly. How they grow, again, depends on their energy source, if they have all of the nutrients required that they need to grow, if they're at the right pH, and our temperature of our bodies is just pretty much prime for most bacteria, and so they really like our body temperature. Some require oxygen or no oxygen, so meaning an aerobic environment or an anaerobic environment. So what I'd like you to do now is if you could cut and paste this particular animation into your browser and watch that and just kind of see how fast these bacteria can actually replicate. You'd be kind of amazed at how fast they do that. So why don't you do that and come right back. Also on Edline, and after you read this, I'd like you to go back to that. There's a link on other ways that bacteria can exchange their genetic information, because we know when they replicate by binary fission, they're making exact copies. So if bacteria evolve, they have to do it by way of exchanging genetic information with another bacterial cell to get something different than they had at the beginning. And bacteria can do this in 3 different ways: conjugation, transduction, and transformation. So I've kind of highlighted what those particular items are here. The textbook doesn't really have a lot of information on this, but it's an important concept. So what I'd like you to do is jot this down in your notes and then go back to Edline and view those animations so you can kind of see what those differences are. And you want to feel comfortable with those so that if we were to make videos on any one of these 3, that you and a potential partner could do that. And then the last thing that we'll talk about today is how prokaryotes protect themselves. They can produce slime layers or endospores, and I've put another animation here that you can watch and cut and paste in that. And that's just in harsh environments. They can actually kind of hang out, and it's almost like hibernation to some extent. As far as endospores are concerned. Slime layers are on our teeth, and so when you go to the doctors, or the dentist I should say, they're trying to scrape that stuff off so that you don't end up with cavities. So that's their way of protecting themselves. So that's bacteria in a nutshell. Hope you've enjoyed the presentation, and I'll see you guys soon.